So I'd like to start by acknowledging the traditional owners and custodians of the land on which we meet today. That's the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present. It's a little bit worrying introducing Simon today because he might get the idea that he's can leave the lab now. See, Simon's been around since the inception of our laboratory, and he's got more papers than I can remember, albeit I do have a bit of dementia, so that statement's not as impactful as it otherwise could be. But Simon's been instrumental in setting up so many of our animal models, and he's helped countless numbers of people, trained countless numbers of people, and he's very much the go-to person for anything to do with LCMV, not just for the Institute, but also for the whole of Australia. He designs the experiments, he performs the experiments, he analyses and interprets the data, and then he writes the conclusion. So he's incredibly important to our laboratory, and indeed, if you were to pluck me out of the lab and not replace me, I think Simon would seamlessly take over running the lab. And if you were to do the reverse and take Simon out of the lab, I think that I would decompensate and everything would fall apart. One of the most amazing characteristics of Simon is that he can, and sometimes a bit annoying, is he can never make a statement as a matter of fact. He'll always challenge you and say, well, where's the evidence? I want to read the paper. And this really sort of underscores his scientific integrity, so much so that I think if you were to put him in Mayer Seller's lab or Joseph Penninger's lab, they would stop publishing. But herein lies one of the biggest conundrums for me, because this same fellow will take every single permutation and combination of vitamin supplements for their anabolic effects, and he's got the most bizarre concoction of high protein, extremely low carbohydrate, intermediate fats, low on the saturated, high on the monos, and with a sprinkling of perhaps a little bit of fibre or something, for their potential anabolic, catabolic and ketotic effects. And I'm pretty sure in another life you could easily have been a Russian Olympic athlete. And I'm pretty sure that you were behind Steve Danks's thymosin scandal at Essendon, particularly because you barracked for Geelong. But despite that major aberration, Simon, you're a phenomenal scientist, and you cannot leave the lab until you work out how you can manage it remotely.
I guess, thank you, Mark. I'm not sure if I sort of live up to most of the things that you said. I definitely don't live up to all the dietary things that you said. Okay. So throughout evolution, immunity has developed to recognise and eliminate infectious pathogens, whilst also developing tolerance to self in order to prevent autoimmunity. When autoreactive immune cells, called T cells, do recognise proteins within our own body, there are inhibitory pathways that are activated to repress their function and reduce damage to the host. When these inhibitory mechanisms are not strong enough, however, to overcome the activation of autoimmune cells, then this can lead to disease and even death on the left-hand side of the slide. Alternatively, a complex interplay between immune activation and inhibition exists during a chronic infection, striking a balance between control of the pathogen and host preservation. Activation of these inhibitory pathways in this situation is a compromise that is made by the host and allows persistence of the pathogen whilst preventing fatal collateral damage caused by systemic immune activation. And it is this complex interplay that occurs during chronic viral infection that I'll be telling you about today, and in particular I'll tell you how cell death shapes T-cell immunity. So one of the main players in our immune system is the CD8 T cell, which is capable of recognising and destroying pathogen-infected cells. Each T cell recognises a specific protein or antigen from the pathogen via its T cell receptor. CD8 T cells function in three main ways upon recognition of cognate antigen. and that is their ability to produce cytokines. They produce many cytokines, but two of those are TNF and interferon gamma. They also have the capacity or the job of killing infected cells, or a cytotoxic capacity, in order to clear the pathogen. And importantly, they also have another function, and that is to proliferate, and that is make copies of themselves so that they can take over and kill all the infected cells to clear the pathogen. If there is constant antigen, however, such as in a chronic infection or autoimmune condition, then this strong inflammatory cytotoxic state can cause serious collateral damage to the host. During an acute infection with a virus, such as the common cold, as we've all experienced, infection leads to viral replication, which is shortly followed by T-cell expansion. And this T-cell expansion quickly leads to pathogen clearance and clearance of infected cells. And once antigen is gone from the system, we see T-cell death, and this is called the contraction phase. And that's the T-cells then going back down to very low numbers to create a memory pool of cells, as they are no longer needed in such large numbers. For my talk today, I'll be speaking to you about a virus known as LCMV, and for my talk in particular, I'll be referring to the glycoprotein and nucleoprotein of the virus, because components of these are what is recognised by the immune system. Now, during a chronic infection, we have a very different scenario that occurs. And whilst we still get infection with the virus, and we see a massive expansion or replication of the virus, because there is so much virus in the system, or so much antigen in the system, we see a scenario where whilst the CD8 T cells expand, they're not expanding to the same level as what we see in acute infection. And as the virus persists, we actually see a drop in T cell number, and this is associated with T cell death, which, as I said to you earlier, should only really occur in the absence of an infection or pathogen once it's cleared. And this overall leads to limited control of the virus, and the infection persists. This phenomenon, this is often associated with a phenomenon known as T cell exhaustion, where these T cells that are around don't have a great degree of function. They've got low functionality in those three main areas that I just described to you. And this phenomenon has been described in many human infections, such as HIV, hepatitis B, and hepatitis C. And it was actually first described in LCMV before it was recognised in these human infections. Now, T-cell exhaustion is caused by prolonged exposure to antigen, and this is the main inhibitory mechanism that I alluded to in my first slide to reduce excessive immune-mediated damage. Here you can see an exhausted T-cell interacting with an infected cell. And the main driver of T-cell exhaustion really is TCR signalling. More importantly, it is the strength of the TCR signal, which is determined by how strongly the TCR actually recognises the antigen that is being presented on the infected cell. As a result of this, there is an array of inhibitory receptors that are then expressed on the CD8 T cell. And these do include PD-1, which has been very well characterised in both chronic infections as well as cancer. CTLA-4, again, recognised in both those conditions with therapeutics that have been developed to target both those pathways, as well as LAG-3. Another hallmark of an exhausted T cell is a decrease in the capacity of those functions of the CD8 T cell that I mentioned before. So they have a decreased capacity to produce cytokines such as TNF and interferon gamma, a decreased capacity to kill infected cells via perforin and granzyme, and they also have a defect in their ability to proliferate or make copies of themselves. So by comparing the immune response in mice infected with an acute strain of LCMV, known as Armstrong, with a chronic strain of LCMV, we're able to get clues as to how and why we observe a loss of high-avidity T-cell clones during chronic infections such as HIV, hepatitis B, and hepatitis C. The mouse is the natural host for LCMV, and so is a nice model to examine the overall immune response to this pathogen. When a black 6 mouse is infected with an acute strain of LCMV, we get a massive CD8 T cell expansion with a pool of cells that recognises an array of LCMV epitopes. And this can be enumerated here. So I'm showing one of the epitopes that the immune system recognises, which is nuclear protein NP396. And we can see that you can get around 5 million CD8 T cells in the spleen of one mouse recognising this one epitope. Whereas when we compare to a chronic infection, that is with LCMV docile, whilst we still do see an initial expansion of the CD8 T cells due to this high antigen load, we see an early deletion of this high affinity NP396 population, where we see several logs less of this one epitope. And again, this is most likely because this epitope has the NP396 cells have the strongest affinity for this epitope and also have the greatest function. And so this is the population that is most likely to cause the greatest degree of damage in the host. So that's why we think that this is occurring. And so it's a really nice model to have a look at what mechanisms are occurring and what cell death pathways might be activated to cause the deletion of these high affinity T cell clones. Just in terms of affinity, where there's a hierarchical breakdown of the affinity of the epitopes that are recognised by CD8 T cells in LCMV, the highest affinity being NP396, the second being glycoprotein or GP33, and behind that is subdominant epitopes. What has been recognised is that the greatest degree of death is seen in the highest affinity T cell clones, and some also occurs in this GP33. Now, the cells that are left over during this infection, as I mentioned earlier, are termed exhausted, and these have defects in their cytokine release, their cytotoxicity, as well as proliferation. And so, as part of my project, we were interested in looking at T cell attrition during chronic LCMV, and we hypothesised that the loss of CD8 T cells during chronic LCMV is mediated through programmed cell death pathways. In particular, we wanted to determine the role of cell death in the attrition of LCMV-specific T cells, and to do this, we looked at intrinsic apoptosis, extrinsic apoptosis, and necroptosis. Furthermore, we also wished to determine the role of T regulatory cells in shaping CD8 T cell responses to LCMV. Apoptosis itself is a highly conserved form of cell death that can be divided into two different arms: intrinsic and extrinsic. Extrinsic apoptosis is induced on the left-hand side here by the ligation of a death ligand with its death receptor, which includes, ligands include TNF, FAS, and TRAIL. This signal is transduced by the activation of Caspase 8, and then activation of cleavage of downstream execution of caspases, and leads to death of the cell. Intrinsic apoptosis, on the other hand, can be induced by a variety of different signals and include stress, cytokine withdrawal, and cytotoxic agents. Depending on the amounts of pro-apoptotic and pro-survival proteins in the cell will determine whether or not this pathway is activated. If it does happen that this pathway is activated, then Baxin will go to the mitochondrial membrane and will puncture this organelle to release cytochrome C and again lead to downstream cleavage of executioner caspases and death of the cell. Now, looking at the main players of intrinsic apoptosis a little bit more closely, we have Bim, Puma, and TBID across the top here, and these cells act to antagonise all of the pro-survival proteins, that is to block their activity on Baxin. We also have Bad and Noxa, and these antagonise two different arms. In particular for my talk and for our research, we're interested in both Bim and Noxa. The reasons for this is that both Bim and Noxa are upregulated upon TCR engagement, and in particular, Bim is what is responsible for the T cell death seen in the contraction phase of an acute response, and so we thought this was a likely candidate for causing the death of cells during chronic infection. And Bim expression has also been associated with exhausted or the death of T cells in patients infected with chronic hepatitis B. We also want to look at Noxa, not only because it's upregulated in response to TCR, but because it antagonises MCL1, which is a very important pro-survival protein for immune cells, and it's also involved in T cells' response to IL-7 and IL-15, which are both very important T cell cytokines. So for the work I'm going to show you today, we used mice that had conditionally deleted these proteins. For this, we used a Cre-Lox conditional system, and for this, we used granzyme B-Cre. And this allowed us to have deletion of our target gene upon granzyme B expression, so when the CD8 T cell is recognising an infected cell. And this deletion, as I said, is upon TCR activated. And so this is a really nice tool that allows us to temporally and spatially delete our genes or proteins of interest at the time that we want to see the effect. And so the purpose of my talk, there are a couple of different genotypes of mice, but to make things a little bit simpler, whenever you see a gene or a protein that's next to a flox symbol, there's been no deletion occurred. And whenever there's a delta symbol, that means that gene has been, or protein has been deleted. Okay, so this work is based on work that was begun by a past PhD student in the lab, Jesse Toe, and I'll be presenting some of that work today. Now, this graph here is showing the fold difference in CD8 T cells recognising either GP33 or MP396 compared to wild type during a chronic LCMV infection. Now, this is done by using things we call tetramers, and these specifically bind to the TCR of either GP33 or MP396, recognising T cells. And you can see here that the loss of Bim leads to a 5-fold increase in the numbers of these GP33 specific T cells, whilst deletion of Bim actually leads to the rescue of between 25 and 30-fold times as many MP396 recognising T cells compared to wild type. Now, when we look again, when I remind you about the affinity hierarchy, we really are seeing a preferential loss of CD8 T cells in a wild type mice, and therefore a preferential rescue with T cells with a high affinity for MP396. So it really does seem to ring true what I said earlier about we are potentially losing the cells that have the highest affinity for antigen and those cells which we know have the greatest function. So now that I've been able to show you that we can rescue these high avidity T cell clones, it's really important to make sure that these cells that we're rescuing actually have function. Because we could only be just rescuing them from dying, and they could just be the living dead, so to speak. So one way that we can look at the function of these T cells is to look at their cytokine producing capacity. For this, we can perform a in vitro or ex vivo restimulation assay, where splenocytes are put in culture with recombinant LCMV GP33 in this case, and then we look at their ability of those CD8 T cells to produce TNF and interferon gamma. Importantly, in the orange and the green here, we see that Bim deficient CD8 T cells are producing not just interferon gamma, but TNF and interferon gamma, which we term polyfunctional T cells. And it's these T cells that are normally the ones that are lost during a chronic infection. So I'm showing you here that not only can we rescue the numbers of these LCMV-specific T cells, but we're rescuing cells that are polyfunctional. I've also shown here mice that are lacking BAX and BAC in CD8 T cells, and you can see that we're seeing a similar rescue between the Bim and the BAX and BAC, which really goes to show that the death of these LCMV-specific cells that is due to intrinsic apoptosis seems to be caused by Bim alone within this pathway. So another really important readout, and arguably the most important readout for the function of a T cell, CD8 T cell, is its ability to kill infected cells, because without that, there's no point having them around. And so for this, what we're able to do is we can get EL4 cells, which are a lymphocyte mouse cell line. We can pulse them or add recombinant MP396 peptide so that these cells will start to express it on their cell surface, or an irrelevant adenoviral peptide to show any background level of killing in the assay. We can then differentially label them with CFSE so that we can differentiate these cells by flow cytometry. And these cells were then injected intravenously into mice at 21 days post-infection. And what this will do is it will be an in vivo assay that shows the cytolytic capacity of endogenous MP396 specific CD8 T cells. So the really true way to look at whether or not these cells are functioning properly. And now when we look at this, we see that the cells that we have rescued are retaining their ability to kill their target cells that are expressing MP396, shown by both Bim and Noxa Bim. And almost all of the cells that we put in there at day 21, that when we take them out, almost all of those cells are gone. And that's shown here by, if we had 100% here, that would be 100% of cells that we put in have been killed. So it really does look as though we are retaining not just the cytokine producing capacity, but the cytotoxic capacity as well of these T cells. So now that I've shown you this, the really important question for us was to whether or not these T cells are having an effect on the viral load in these mice. So now when we look at the viral titers of this mice, there's quite a lot of data on this slide. But what we can start to see is, compared to wild type in the black, mice that have CD8 T cells lacking either Bim or Noxa and Bim have a very strong trend towards clearance. And already in the liver and spleen, we're seeing the Bim mice that we have seeing significant drops in viral load. And this is at 60 days post-infection. And so we really are seeing that these increasing numbers of cells are starting to have an effect on the system. And maybe it is the case that if we were to take these mice out a little bit longer, perform these experiments again, perhaps it would lead to complete control of the virus. But the other option here is maybe there's something else that we haven't quite noticed yet. And to do this, I went back and looked at the numbers of these cells again. Now, when we go back and look at the total numbers of these either GP33 or MP396 specific CD8 T cells, and we compare them to the numbers of the same cell population in acute infection for GP33 or MP396, taking note that these graphs are on a log scale, you can see that we're still not quite reaching the levels that we're seeing in acute infection for both these cell populations. And so maybe it's something else that we haven't quite addressed yet. Now, looking at this hierarchy once more, I've been able to show that we can rescue cells with cytokine producing capacity, cells with cytotoxic capacity, but what about proliferation? So the ability of these cells to make more copies of themselves. And maybe this proliferation is lacking a little bit, and that's why we're not quite reaching the level seen in acute infection. So the way that we can look at proliferation is by use of an in vivo proliferation assay, and this can be done by the injection of a compound called BRDU. This can be injected into the mice the night before you sacrifice the mice, and then when you take the splenocytes the next day, you can actually stain for this intracellularly, and cells that stain for BRDU cells that have recently proliferated. Now you can see quite clearly here that there appears to be a striking defect in the ability of these cells to proliferate. So in the orange here, Bim deficient CD8 T cells or Noxa Bim deficient CD8 T cells, they have several-fold reduction in their capacity to proliferate compared to the relevant controls. So here we may have answered our question, and that perhaps the reason why we aren't completely rescuing virus-specific T cells compared to the acute infection is due to a proliferation defect. So to address what might be inhibiting the proliferation of CD8 T cells in the Bim mice, we had several candidates that have been described in the literature. PD-1 is a pathway that is already being targeted clinically as an immunomodulatory treatment for many cancers with encouraging results. IFNA, which is the receptor for type 1 interferons. This pathway has already been blocked with encouraging results in other research looking into chronic infections. Could it also be that other death pathways are playing a role in killing off a population of cells that is responsible for proliferation or could help with the proliferation in these Bim deficient mice? Or could potentially CTLA4, which is another inhibitory pathway that's being targeted in cancer immunotherapy. And the other option is, could it be cytokines that we already know are responsible for inducing senescence rather than proliferation? So the questions were, are these pathways inhibiting proliferation in any way? Can we antagonise these pathways to restore proliferation? Or alternatively, is there an intrinsic factor which haven't been able to identify yet that these Bim deficient T cells, meaning that these Bim deficient T cells have a proliferation defect? The last possibility is that maybe there's multiple pathways that are being agonised that are leading to this proliferation defect, and maybe there's another cell known as a regulatory T cell, perhaps that is agonising multiple pathways here and leading to a proliferation defect. So when we look at PD-1 expression on our LCMV specific T cells, we found a relative disconnect with the literature that despite expressing high PD-1, shown with the solid black line and the green line here, being with cells both Bim deficient and sufficient, we can see that we actually have very, very high expression of PD-1 on our rescued T cells, and the literature would suggest that if a T cell is expressing high levels of PD-1, that this T cell is exhausted and cannot function. However, I've just shown you that these very same T cells do have function. They can produce cytokines, they can kill target cells. And so whilst they still have function, we thought that perhaps even if we thought that maybe we can still antagonise this pathway by blocking PD-1, that perhaps we can encourage these cells to start proliferating a little bit more, and hopefully that might be able to tip the cells over the edge to be able to control virus a little bit better. So here, we targeted the PD-1 pathway by blocking the interaction between PD-1 and its ligand PD-L1 using a monoclonal anti-PD-L1 antibody. Mice were infected with LCMV and then were treated every second day from days 10 through to sacrifice on day 21 with either anti-PD-L1 or an isotype control. Now, despite PD-1 blockade, we did not see an enhancement of proliferation in our Bim deficient T cells, and in fact we saw a decrease in the proportion of activated T cells proliferating. This, however, might be explained by the expansion of non-specific virus T cells that have begun cycling in the presence of anti-PD-L1, and that perhaps might have diluted the T cell pool being analysed. Next, we wanted to see if blocking type 1 interferon signalling might help with reversing the proliferative defect. Here we used anti-IFNA antibodies, which block the interaction of interferon alpha and interferon beta with their receptor IFNA. So for this, mice were again infected with LCMV Docile, and we treated mice with either anti-IFNA or an isotype control at days 10, 12 and 14 before sacrificing the mice on day 17. And you can see quite clearly that blocking type 1 interferon does not restore the proliferation defect in these Bim deficient T cells. And again, we're consistently seeing this decrease in the general ability of these Bim deficient T cells to proliferate. So it wasn't type 1 interferon. So they're the things that we've tried so far to restore this defect. And so in summary of my talk so far, when we compared to wild-type mice, I've been able to show you that mice that contain T cells lacking both Bim and Noxa, they have enhanced survival, enhanced function. They do still have poor proliferation, but we see an enhanced ability to control virus. What we think is happening in these mice to explain the increases in function is that despite the highly inhibitory environment seen in chronic LCMV, is that we are rescuing T cells that in fact have a higher avidity for their cognate antigen, and it's these cells that have the enhanced cytokine production and cytotoxic capacity. In other words, highly functioning LCMV-specific T cells are being deleted early on in infection, leaving behind only T cells that are relatively dysfunctional, and these are the cells that people in the literature often call exhausted. The answer to working out the proliferative defect could still lie in other inhibitory networks. It could also be an intrinsic defect, which I will be addressing by performing T cell transfer experiments, or it could also be other death pathways that are killing off cells with proliferative potential. So looking at other cell death pathways, I'll now be talking to you about extrinsic apoptosis and other cell death pathways downstream of these receptors. The prototypic receptor for extrinsic apoptosis is TNF-R1, which is shown here. However, this can be misleading as ligation of TNF with its receptor actually drives NF-kappa B mediated pro-survival and pro-inflammatory cytokine production. It's only when NF-kappa B signalling is inhibited by SMAC mimetics or other mechanisms that other outcomes are possible. And we can sometimes see the formation of this complex of this complex shown here in the middle, and this can occur if Caspase 8. And what we can also see when this complex is formed, that we can have Caspase 8 homodimerization, and we can have the induction of cell death or extrinsic apoptosis. Depending on the abundance of Caspase 8, however, death can also occur by a different mechanism that involves RIP kinase 1 and RIP kinase 3. And this leads to the phosphorylation of a protein called MLKL, and the formation at the membrane and the formation of a complex of these MLKL proteins at the membrane and can lead to an inflammatory type of cell death known as necroptosis. Now, both of these pathways could very well be active during a chronic LCMV infection due to the persistent inflammatory environment, and these may be contributing to the death of LCMV-specific T cells and maybe the population of cells that are proliferating. So at this time, there was a new form of cell death, which was necroptosis. This was several years ago now. It was a very hot topic in the area of cell death, and we were lucky enough to have mice available to us within the institute. And we wanted to see if this type of cell death was responsible for the death of LCMV-specific cells. Using RIP3 knockout mice, we found that that cannot undergo this form of cell death, we found that there was no difference in the numbers of GP33 or MP396 specific CD8 T cells at any of the time points that we looked at from day 8 right through to day 105 post-infection. I also did not see any difference in the clearance kinetics of LCMV at any of the time points that I looked at, right from early time points at day 3 right through to 105 days post-infection. So it really did see as though necroptosis wasn't affecting the death of these LCMV-specific T cells. And now to address whether extrinsic apoptosis contributes to the death of LCMV-specific T cells, we again were lucky enough that John Silk within the Institute already had these mice, and he kindly gave us some mice with breeding pairs. So these are mice that were lacking both Caspase 8 and RIP3. So these mice are both unable to undergo cell death via necroptosis or extrinsic apoptosis downstream of death ligands such as TNF, FAS, and TRAIL. Now looking at 28 days post-infection, you can quite clearly see that extrinsic apoptosis does contribute to the death of GP33 specific T cells. As we can see, a twofold rescue in the number of these cells. However, we did not see rescue in the MP396 population, this high avidity population. But as before, we wanted to know whether or not these cells that we can rescue here still do have function. So now performing the same re-stimulation assay as I described to you earlier. We have exhausted T cells often only express interferon gamma. As I said to you before, that it's only polyfunctional T cells that are the ones that really have high function. And we can see that in these mice that cannot undergo extrinsic apoptosis, and we're rescuing cells, that we're only rescuing cells that can produce just interferon gamma. So we're not rescuing the polyfunctional cells that we were able to earlier. And it's highlighted here. You can see that there is quite a strong disconnect between the type of cells that we've been rescuing. So again, I wanted to look at the ability of these cells to kill. This time, I didn't do an in vivo proliferation assay, but using a surrogate marker, we can, from the same restimulation assay, we can look at the expression of granzyme B, so granzyme, which is responsible for killing infected cells. And we can see that these cells that we are rescuing that are recognised in GB33 do still retain this cytotoxic function. So as compared to the Bim mice, we're only rescuing a very small portion of T cells, albeit significant, and they still do have function. So we still wanted to see whether or not rescuing these cells was impacting the viral load of these mice. And it's quite clear that we were unable to. And that makes sense because, again, we're only really rescuing cells that have poor function and that are probably really still exhausted. So in summary, for the second part of my talk, I've shown that necroptosis does not contribute to the loss of LCMV-specific T cells, and also that RIP3 deficiency does not affect the clearance of LCMV. I also showed you that extrinsic apoptosis does contribute to the loss of LCMV-specific T cells, but is only capable of rescuing GB33-specific cells and not the high avidity MP396 cells. The rescued cells have poor function. The rescued cells have poor function, and these cells that are being killed most likely through an unintentional death receptor killing rather than an attempt by the host to reduce collateral damage from highly functioning CD8 T cells during a chronic infection. Now, taking you back to this slide that I showed earlier, another reason that we could be seeing poor proliferation in our Bim deficient T cells or any T cells during a chronic infection is a suppressive function of T regulatory cells, also known as Tregs. These cells could be acting through multiple pathways to inhibit CD8 T cell responses to a chronic infection. T regulatory cells' job is really to fine-tune immunity. Through a variety of mechanisms, they allow bona fide immune responses to infectious pathogens, whilst also suppressing inappropriate immune responses to things like self-antigens. And they do this by acting on conventional CD4 and CD8 T cells. So for this part of my talk, I'll be presenting work that has been done on a collaborative basis with Daniel Gray and Caspase within the Institute. And they work on T regulatory cells, and they're interested in the role of Caspase 8 in these Tregs. They're interested in finding out whether or not extrinsic apoptosis plays a role in Treg homeostasis. They were also interested in if this pathway regulates Treg survival during chronic viral infection. And we were also very interested in this collaboration because we'd be able to look at the potential role that Tregs play during a chronic LCMV infection. For this work, we used a mouse that had a conditional deletion of Caspase 8 only in Tregs, which is denoted by this Caspase 8 FOXP3 symbol here that you'll see for the remainder of my talk. Now, when Caris came to us with the data, she showed us that at homeostasis, extrinsic apoptosis appeared to be important for restraining Treg numbers in the spleen. You can see here both by proportion and by total number that Caspase 8 FOXP3 mice had much more Tregs in the system. And we wondered that if these mice had more Tregs, then perhaps the question was, would immunity to LCMV be even further suppressed by the greater number of Tregs in the system? But what we noticed immediately that was upon LCMV infection, these numbers drastically dropped, both by proportion and by total number, comparing Caspase 8 FOXP3 mice uninfected or infected mice. So something to do with the inflammatory milieu appeared to be changing the effect of Caspase 8 loss in these Tregs. So With this in mind, so with these findings in the Tregs, and our finding that there's a differential requirement for Caspase 8 during homeostasis and infection with chronic LCMV, we had a couple of questions. We wanted to know if reduced Treg numbers shape our T-cell immunity, and in particular, does the drop in Treg numbers in Caspase 8 FOXP3 mice enhance LCMV-specific T-cell responses? We also wanted to know whether the drop in Treg number enhances T-cell proliferation. Also, if the drop in Treg numbers in these FOXP3 mice release the suppression of inappropriate T-cell activation, in other words, non-LCMV responses. And also wanted to know if this differential requirement for Caspase 8, whether the inflammation of chronic LCMV is driving another form of cell death in the absence of Caspase 8. So in the first couple of experiments, we wanted to have a quick look to see if the drop in Treg numbers was affecting the numbers of LCMV specific CD8 T cells. And for this, we just looked at GP33 recognizing cells. And indeed, we're seeing a 2 to 3-fold increase in the number of these cell population in the FOXP3 mice compared to wild type controls. As we were seeing an effect of the drop in Treg numbers, we immediately wanted to see if there was any effect on the proliferation of these cells, or just proliferation in general. And indeed there was. This time I decided to use KR67, which is an endogenous marker of proliferation. And you can see on this histogram here that in the FOXP3 mice, which are in blue, we have a much greater proportion of cells that are proliferating compared to wild type. This is occurring in both the CD8 and CD4 compartment of these mice. And just looking overall at activation, because we know that Tregs are responsible for either allowing activation or suppressing the activation of T-cell pools, we can look at CD44 as a general marker of activation, and you can see that we are also seeing enhancement of T-cell activation across the board, seeing quite a massive increase in the activation of these both CD8s and CD4 T-cells. So one important way to look at the effect of Treg numbers in a mouse is to look at the ratio of Treg cells to activated T cells, as it really is the balance of Treg suppression versus activation. You can see here that in the FOXP3 mice, we're seeing a much lower ratio of Tregs to activated cells, and this really means that there is a lower number of Tregs per activated cell in these mice. Meaning, and perhaps with this sort of ratio and having a much greater number of activated cells, perhaps we might also be seeing a degree of collateral damage that is occurring during this chronic infection. Now here I'm showing the levels of a liver enzyme in the serum of our mice that is liberated upon liver damage or death. And you can see that the levels of AST are significantly higher in the FOXP3 mice here in the blue boxes. And that is consistent with the enhanced number of LCMV-specific T cells in these mice, but also potentially an enhancement in the number and activation of non-LCMV-specific cells as well. So to address this question of whether the drop in Treg numbers is leading to the activation and expansion of non-LCMV-specific T cells, I performed this experiment here. So for this, I used tetramers that bind to CD8 T cells that do not recognise LCMV. These cells, if expanded and activated, could be causing collateral damage through activation and expansion of non-LCMV-specific CD8 T cells. And these are the ones that could be causing the degree of damage and the increase in the liver enzymes that I've just shown you. And what we can see here is that in Caspase 8 FOXP3 mice, we have 2 to 3-fold as many of these non-specific LCMV cells that are activated, and this number is per mouse. And so we are seeming to be getting an increase in the activation and number of these cells that could be causing collateral damage. So in order to test this, we wanted to see if these mice might succumb to an autoimmune-like condition if we let the infection go for a longer period of time. You can see here on this survival plot that when we infected FOXP3 mice and wild-type mice with chronic LCMV, they began to get sick around 7 weeks post-infection. These mice had severe weight loss, they had ruffled fur, and they needed to be euthanized. They also had multi-organ immune infiltration. Now, this really does highlight the role that Tregs appear to play in suppressing non-pathogen-specific T-cell immune responses during chronic infections such as LCMV, and really confirms that during chronic infections, if the immune response is not inhibited in some way, then the host immune system can cause fatal immune activation. Now you can see here, looking at the viral loads of these mice from the long-term experiment, that FOXP mice, they were actually able to survive, and these are the triangles shown on the right here. These mice made it out to 145 days post-infection. They were actually able to clear virus. You can see then in the blue squares, and these were mice that needed to be sacrificed because they were sick. We see that a number of these mice had begun to control, and these two mice that haven't, they're the two mice that were taken the earliest, and so were unable to control virus at that time. So this again highlights the fine balance that exists between controlling a chronic infection and protecting the host from fatal immune activation. So now that I've shown you the effect of low Tregs in the FOXP3 mice and how Tregs really do appear to be the master regulator between host survival and death during chronic infections, we wanted to find out how these Tregs were dying in the first place. Now, it's known that Caspase 8, it is known when Caspase 8 is inhibited chemically or knocked out by genetic means that necroptosis can occur, and this can lead to the death of these cells. And we wondered if necroptosis might be active for some reason in Caspase 8 deficient Tregs during infection, but not during homeostasis, because I've already shown you that at homeostasis, Caspase 8 deficient Tregs have higher numbers, not lower. So for this, I took the spleen and lymph node from Caspase 8 FOXP3 mice or control mice, sorted these cells into either Tregs or conventional CD4 T cells, and then subjected them to Western blot analysis with the help of the Western blot guru in our lab, Michael Stutz. Now you can see very clearly that as opposed to an uninfected mouse where you almost see no MLKL, you can see that upon LCMV infection, there's a massive upregulation in the amount of MLKL that's expressed in these cells. And we wondered if whether or not this is what might be mediating the death in these cells and was allowing these cells to be killed during infection and not during homeostasis. And possibly it's the inflammatory milieu that occurs that's inducing the upregulation of MLKL. So to confirm this and see if blocking this pathway led to the rescue of this Treg death and reversal of fatal immune activation, we sought mice lacking both Caspase 8 and MLKL. Now, when we infected these Caspase 8 MLKL mice and looked at day 8, we already saw that we were able to reverse the Treg to activated Treg ratio. Caspase 8 MLKL mice here are in the green. And so it appears as though blocking the death of Caspase 8 deficient Tregs mediated by MLKL-driven necroptosis may indeed rescue the fatal phenotype that we saw in the FOXP3 mice. And when we looked at our LCMV and non-LCMV specific readouts, we could see that upon re-stimulation and comparing the cytokine production in wild-type mice, in the red here, compared to our FOXP3 mice, which with their LCMV specific responses had greatly enhanced numbers of cytokine production. And now when we compare to the Caspase MLKL in the green, these appeared to be much more like wild-type. When we looked at CD44 expression, that marker of activation, we see in the green that the levels of pan activation appeared to be much more similar to wild-type than they are the FOXP3, which still had massive amounts of CD44 expression. And now when we look at proliferation, again, in these same mice, we see that the proliferation in the Caspase 8 MLKL looks a little bit more like wild-type than it did in the Caspase 8 FOXP3 mice. We're also able to show the functional result of rescuing these Treg numbers was restored, shown here by liver AST levels in the serum, indicating that collateral damage appears to be rescued. So it's not just that the cells look back like wild type, we don't appear to be seeing the collateral damage that we see in the Caspase 8 mice that leads to their death. So to summarise my talk today, I've shown you that the BH3-only protein Bim is responsible for driving the death of highly functional LCMV-specific CD8 T cells with high avidity for antigen. I've shown you that rescuing these cells leads to enhanced viral control, although complete control may be inhibited by a proliferation defect. The fact that these mice, however, do not become overly sick or die, even though we've rescued highly functioning CD8 T cells, suggests that the immune system may be dampening T cell immunity beyond what is required to preserve the host. These findings therefore provide evidence that targeting this pathway may be a potential therapeutic intervention in chronic infections or even cancer immunotherapy that may be safer than therapies that lead to general immune activation. Furthermore, I've presented data to support a new model of T cell exhaustion, and that is that highly functioning pathogen-specific T cells are deleted early on in infection, leaving behind them T cells that are relatively dysfunctional and often termed exhausted. I've also shown you today that extrinsic apoptosis mediated by Caspase 8 drives the death of LCMV-specific CD8 T cells, but that this rescue is only seen in lower affinity GP33 specific T cells with poor function. I've also shown you that Treg cells have a differential requirement for Caspase 8 during homeostasis and chronic infection, and I've highlighted a critical role that Treg cells play in suppressing non-pathogen specific T cell responses during a chronic infection, and that this fine balance, if tipped towards activation, can lead to fatal immune complications. Lastly, I showed that the death of Tregs in the absence of Caspase 8 is driven by MLKL-mediated necroptosis. So that just leaves me to thank my supervisor and mentor, Mark Pellegrini. His support for me over the years is indescribable. He is truly a pleasure to work with, and he's not only an incredibly intelligent supervisor who is successful in attaining funding, but he is also highly democratic and allows his students a huge degree of freedom with the direction of their projects. I'd also like to thank Jesse Toe, past PhD student in the lab, for his involvement in this project, as well as Daniel Gray and Karis Tay, obviously, for their involvement in our collaboration on the Treg Caspase 8 project, and hopefully that collaboration continues. I'd also like to thank Andreas, Philippe and Warren for mice. Bioservices, obviously, a project like this, and I guess all projects in our lab are highly dependent on mouse work. So our techs, Christy and Carolina, as well as a massive thanks to Liana, who used to be an animal tech and looked after our mice for years and years, and we've actually managed to get her onto our payroll now, as well as my committee, and also, of course, my family and friends, and I'm happy to take questions.
Now, we have to be out of here by 4 o'clock, so we've only got time for a couple of questions, but obviously we'll be outside if you've got more questions for Simon. So we'll take as many as we can before we get kicked out, which will be in 4 minutes.
Axel?
Got a question to the first part of your talk. You said that you have a lot more antigen-specific cells in the Bim knockout, but you didn't seem to get any immune pathology. So I was wondering whether, as a proportion of the antigen-specific cells, you still had as much exhaustion or as little interferon gamma, so that the increase in interferon gamma was just a reflection of the overall increase in cell numbers. I'm not sure if I expressed myself correctly, but what I wanted to say is, do you actually get a rescue of function, or do you get just a rescue of numbers?
So it's both. So if you look at the Bim mice, you hardly see any CD8 T cells that recognise MP396 that produce TNF and interferon gamma. But what we do see in the Bim mice is you're rescuing cells that are producing both TNF and interferon gamma. So we're rescuing cells that are polyfunctional, which we don't actually see in a wild-type mouse. And we're also seeing increased numbers of both single positive and double positive T cells.
Any idea why you don't get pathology?
No, I'm not sure why. I mean, the mice don't look exactly like wild type. They look like a little bit sicker in the first week or two, but they seem to recover. And yeah, like I said in my talk, then maybe the immune system's overcompensating a little bit by deleting these cells, because they can show by rescuing them that they actually don't succumb to that immune activation. And that does happen because you can see it when you get rid of Tregs.
Puzzled by the Caspase 8 FOXP3 deleted line that you were using. So you show that if you lose Caspase 8, you get a more immune pathology. But then if you remove MLKL, so you're removing another death pathway, you then seem to rescue that effect. Whereas I would have expected you to get more immune pathology because you're blocking another mechanism of death.
Yeah. So good question there. So when you have just the Caspase 8 deficient Tregs, when you see that drop in Treg number, and we see that ratio changing such that you're getting fewer Treg cells suppressing the potential activation of non-pathogen specific T cells, which I've shown will occur. So we seem to be getting more like an autoimmune type thing where you're getting T cells that are recognising the host and causing collateral damage. Now what we did when we then used had Tregs that were deficient in both pathways, we stopped those Tregs from dying by necroptosis. So that ratio is now back up to what we're seeing in wild type, where we're suppressing the activation of those self-specific T cells and reducing that collateral damage.
This will be the last question.
So just going back to this part where you look at RIB3 knockout and RIB3 Caspase 8 knockout, and you show that there's no involvement of necroptosis in rescuing the proliferation defect that you were seeing in LCMV infections. Did you look at Caspase 1 at all, given that, like they're both HIV and LCMV are both lymphocytic viruses, and HIV is known to kill white blood cells, CD4 T cells at least, by Caspase 1.
Yeah. So I have looked at Caspase 1 knockout mice, so just total body Caspase knockouts at a couple of time points, and I didn't see any overt phenotype. It didn't appear to rescue the numbers of virus-specific cells. I looked at both Caspase 1 knockouts as well as Caspase 1 and 11 knockouts, and didn't seem to be rescuing them, at least in our LCMV system.
So please join me in thanking Simon, and if there's any more questions, we'll go outside.